Today we are going to study about enzymes, and here is the checklist for today's lecture. Firstly, we will begin with an introduction to enzymes. Then we will discuss the discovery of enzymes, followed by the characteristics of enzymes. Then we will study the structure and composition of enzymes. Lastly, we will study factors affecting enzyme activity. So let's begin with introduction to enzymes. First of all, let's talk about what enzymes are. The word enzyme is derived from two words, enzyme, meaning enzyme meaning yeast. It was named enzyme because it was first discovered in yeast. Do you know that in 1878, a scientist named Friedrich Wilhelm Kuhn was the first to use the term enzyme? Enzymes increase the rate of chemical reactions. And did you know how enzymes increase the rate of chemical reactions? Let me tell you how. They lower the activation energy required for a reaction. By lowering the activation energy, enzymes can significantly speed up the rate of chemical reactions. Now you might be thinking, what is activation energy? Activation energy is the energy required to start a reaction. Let me explain this with the help of our graph. In this graph, you see two lines, one blue and one red. The blue line is lower and the red line is higher. The blue line represents the reaction when an enzyme is used, and the red line shows the reaction without an enzyme. As you can see, the red line is higher and it needs more energy to complete the reaction. In the red line, more energy is required, which is why it is higher. This requires more energy to complete the reaction because the reaction is completed without an enzyme. Below you can see the blue line. Here an enzyme is used to speed up the reaction in the blue line. That's why in the blue line less energy is needed because the enzyme lowers the activation energy, making the reaction proceed faster. So this is the function of enzymes, to lower the activation energy and thereby increase the rate of the chemical reaction. Enzymes as biocatalysts. Did you know that enzymes are also known as biocatalysts? The term bio refers to life, and a catalyst is a substance that speeds up a reaction. Since enzymes are found in living organisms and they increase the rate of chemical reactions, they are called biocatalysts. Characteristics of enzymes. Now let's move on to the characteristics of enzymes. Number one, enzymes are proteins. The first characteristic is that enzymes are made up of proteins, which is why we say that their nature is proteinaceous. If you need more details about proteins, I have created a detailed lecture on proteins. You can watch this lecture by visiting my channel. Enzymes can react with both acidic and alkaline substances. Number 3. Enzymes are not consumed in reactions. They are not used up. Instead, they remain unchanged and can be used repeatedly. For example, in this reaction, you see the blue enzyme combining with the substrate to form a product. After the reaction, the blue enzyme remains the same, indicating that it can be used again without any changes. Number 4. Enzymes are specific. The fourth characteristic is that enzymes only catalyze specific reactions. For instance, we have some biological molecules such as proteins, lipids, and maltose. The enzyme that breaks down lipids is called lipase. Other enzymes such as protease will not perform this function. In the same way, the enzyme that breaks down proteins is called protease. Lastly, the enzyme that breaks down maltose is called maltose. So every enzyme is specific in its function. Number 5. Naming of enzymes. Another important point to remember is how enzymes are named. Enzymes usually end in as, or gen. If the name ends in gen, it indicates the enzyme is in an inactive form. If the name ends in "as", it means the enzyme is active. For example, trypsinogen is an inactive enzyme. For example, lipase and maltase. Enzymes become active from their inactive forms before they start working. The fifth characteristic is that enzyme activity can be increased or decreased. Substances that decrease enzyme activity are called inhibitors, while those that increase enzyme activity are called activators. Now, let me tell you the factors affecting enzyme activity. There are several factors that can affect enzyme activity, causing it to increase or decrease. They are listed below: the pH, temperature, alcohol, substrate, concentration, radiation, activators, inhibitors, coenzymes, and cofactors. All these factors influence enzyme activity in different ways. Structure and composition of enzymes. Enzymes are protein in nature, meaning they are made up of proteins. There are two main types of enzymes: simple enzymes and complex enzymes. Let me explain them one by one. Number one, simple enzymes. These enzymes are made only of proteins. Number two, complex enzymes. They are also known as conjugated enzymes or holoenzymes. These enzymes are made up of two parts, in which one part is protein and the other is non-protein part. The protein part of a conjugated enzyme is called a coenzyme, and the non-protein part is called the prosthetic group. Again, non-protein part are of two types, which are listed below. The non-protein part of a conjugated enzyme can be either organic or inorganic. If it is organic, it is called a coenzyme. If it is inorganic, it is called a cofactor. Let me give you examples. In many enzyme reactions, there's a non-protein part that helps the enzyme do its job. If this non-protein part is a metal ion like magnesium, potassium, manganese, or calcium, we call it cofactor. These metal ions assist the enzyme in catalyzing reactions. Now there's another type of factor called the coenzyme. Coenzymes are organic molecules, usually derived from vitamins, and they bind to the enzyme to assist in its activity. Coenzymes help transfer chemical groups, like hydrogen or phosphate, from one molecule to another during these reactions. To clarify, coenzymes are a subset of factors, while cofactors can be either inorganic metal ions or organic molecules. Coenzymes are specifically the organic ones. Examples of coenzymes include NAD, which is derived from vitamin B3 or niacin, and coenzyme A, which comes from vitamin B5 or pantothenic acid.